Well, Stuart's given you a bit of my career, but really my qualification for speaking to you tonight is partly the time I spent at the department, which used to be called Biology and Biochemistry, that's now part of a larger Department of Life Sciences here up the hill, and for 6 years running this Stem Cell Institute in Minnesota. This looks good, but it wasn't actually occupying the whole building. So in fact, it was a rather smaller enterprise. It was a better paid job, but a smaller enterprise than running this department. Now, in fact, the most important thing I think I really need to say may surprise you a bit, which is that if you look up stem cell research on Google, or if you look in the majority of press reports about stem cell research, what you're looking at is mostly rubbish. I'm sorry to have to say this, but this chap here is pretty widespread around the world, running private clinics and even some relatively distinguished medical complexes. And a lot of people do believe this, which is really completely misleading. The idea is, I've got some miracle cells. This says fat-derived stem cells, if you can read it, but they could be from almost anywhere. And they can turn into anything. And if you've got some disease where part of your body is dying off, we'll inject some of these. They'll grow and repopulate and regenerate the structures that are damaged. Now, this is completely untrue, and it doesn't happen. So most of the, it's not all bogus, it's partly well-intentioned, but with little scientific rationale. So a lot of this so-called stem cell therapy consists of grafting bone marrow from one part of your body to another, which on the whole has no effects. And it quite often consists of buying some cells like this from a company and injecting them into the part that hurts. Now, if you do that, you're injecting cells from one person into another. You're doing an allograft, and that means that all the cells you inject are going to be dead within 48 hours. And actually, people do agree that that is what happens. Sometimes there have been very careful controlled trials, particularly on cell transplants into the heart. And sometimes these trials have shown some positive effects, but in no case do the cells persist and turn into heart muscle. If there are positive effects, they are short-lived, a few months, and they're probably due to some sort of anti-inflammatory effect or immune modulation or some poorly understood function of the cells, of whatever these cells release as they're dying. So I'm not saying it's all absolutely without effect, but I am saying it's all absolutely without scientific rationale. So in no case do the grafted cells rebuild the damaged tissues. So I could stop now and send you all away, and you'd be very disappointed. But there are real stem cells, and they are even to some extent used in therapy. So what is a stem cell? It's a cell, and this shows you a typical stem cell in your body. Stem cells divide to produce more of themselves, so they're an immortal population, which persist typically as long as the organism persists. And they also give rise to usually more than one type of functional differentiated cell. And usually they do it by undergoing a succession of amplification cell divisions. So what that means is there are very few of these in the circumstances where they exist, and there are quite a lot of these. And there are also a lot more dividing cells than there are stem cells. So not every cell that's dividing is a stem cell. In fact, it's quite difficult to identify stem cells in vivo, and I'm not going to really go into that with you. Now, here's an important thing to grasp if you're not familiar with this field, that fitting the definition I've just given you are two basically different types of stem cell. There's what we now call pluripotent stem cells, and these are embryonic stem cells that you've all heard of and you all know are very controversial or used to be very controversial. or they are induced pluripotent stem cells, which are essentially the same, but are made by a method I'll show you in a minute. And these types of cell really only exist in vitro, in culture bottles in the lab. They don't exist in your bodies. And they can be grown forever in suitable media, and they can produce any cell type in the body, probably produce any cell type in the body with the appropriate treatments. So that's pluripotent stem cells. And the other type of stem cell are those that do exist in your body, called tissue-specific stem cells. And these are responsible for maintaining those tissues in your body which continuously turn over in adult life. And I'll give you some examples of those in a minute. So let's look first at the pluripotent stem cells. So the first discovered of these and the best known are the embryonic stem cells. And this is an early human embryo here. Well, strictly speaking, it's a pre-embryo or a conceptus, because not all the cells you see here are going to become the embryo itself. All these cells around the outside are going to become the major part of the placenta. And there's this little cluster of cells on the inside called the inner cell mass. Now, some of those are going to become part of the placenta as well. And actually, at this stage, you can't really tell which cells are going to become part of the placenta and which aren't. In fact, nearly all the cells are going to contribute cells to the placenta. But these inner cell mass cells will also contribute cells to the actual fetus. And if you take these out and put them into culture and give them a witch's brew of the right sort of substances, then you can culture them and they'll grow forever. And this is what they look like. Now, actually, these ones have been colored green. They're not naturally green. That's a genetic trick to make them go green. And these are the stem cells here. And these are not individual cells. Each of these green blobs is a colony of cells. So probably some, maybe a dozen, perhaps 50 or 60 cells in these clusters. And in between, you can see some shadowy gray things. These are what are called feeder cells. These are ordinary tissue culture cells, which are often called fibroblasts, which have been irradiated, usually irradiated to a degree which maintains them metabolically alive, but means that they can't divide anymore. So they produce a sort of nutrient soup, which makes the embryonic stem cells feel happy and keep growing. So this used to be the standard way of growing both mouse and human embryonic stem cells, on these feeder layers of irradiated fibroblasts. Nowadays, you can grow them in completely synthetic media, and the key substances produced by the fibroblasts have all been discovered. And in fact, for clinical grade cells now, you would not want to use, you would want to use completely defined products for growing your cells to try to minimize the chances that they might introduce viruses or other nasties into your patients. And I've already told you that you can make embryonic stem cells develop into just about anything, including heart muscle, neurons, pancreatic beta cells producing insulin. And I'm, again, not going to go into how you do this, but it turns out that what I did for most of my career, figuring out how embryos develop, is absolutely critical in being able to do this, because the protocols for making embryonic stem cells into functional cells involve treating them with a sequence of different media components which mimic the normal set of developmental decisions that those cells would go through if they were part of an actual animal or human embryo. So when I arrived in Minnesota, everybody thought I was very highfalutin pure scientist, but I did try to explain that what I'd done with frogs was partly relevant to what they still did today, because we collectively discovered all the factors they put on their cells to make them differentiate. And here's the other sort of pluripotent stem cell. And this is fantastically important. Actually, the year before I went to Minnesota in 2006, this guy in Japan, by a really brilliant tour de force, one of the best bits of science I've seen in my lifetime, and he did get a Nobel Prize, figured out that you could take ordinary tissue culture cells. These are fibroblasts grown from the skin, could be mouse skin, could be human skin, and you put 4 genes into these cells. And again, I won't explain how you put them in, but there are all sorts of different possible ways of doing that. And these 4 genes were selected because they were known to be important for the pluripotent behavior shown by mouse embryonic stem cells. And actually, it's not always the same 4 genes, but it's about 4, and they all belong to this category of genes known to be important in controlling pluripotency. And you put your genes into your cells, you put them into our ES culture medium, and after a couple of weeks, you've got some colonies, and they'll be looking pretty familiar to you by now. Those are iPS cells, which are just about identical to embryonic stem cells, and they even go green if you put a nanog reporter into them. So you can do everything with iPS cells that you can do with embryonic stem cells. The true potential, though, the real potential, which is as yet very not really realized, or only realized on a very small scale, is that you can make iPS cells from anybody, make from me or any one of you. And that means if I needed a graft which was immunologically compatible with me, you could make iPS cells, make the cells I need from those and inject them, and that would be an immunocompatible graft. Now that sounds great, but the practical and financial obstacles to doing that are enormous. And as far as I know, there's only been a couple of such cases carried out clinically, and they've been done in Japan. And well, iPS cells can do everything ES cells can do, and these are that's some heart muscle that we made, and that's some neurons, and you can even make beating human heart muscle. And one of our people at Minnesota made these about 10 years ago now, so he was very good at making them. And there is some real bona fide scientific rationale stem cell therapy with cells, not with the pluripotent stem cells themselves, but with cells made from them in vitro and then grafted into the patient. And I might say you would never want to graft actual pluripotent stem cells into a patient, because we know one of their properties is that they form tumors called teratomas, and that's very dangerous. So you don't want that. So here are some current projects which have been through a number of smallish clinical trials in the last 10 years or so, and which are all looking reasonably optimistic in terms that they probably do work and will be the basis for quite significant new types of cure in the future. I'm going to show you more detail on the first one. Treatment of diabetes is something I had a significant research interest in myself, and I think this is very interesting, but the number of cases so far is very small, and I didn't want to show that to you. Parkinson's disease, the grafts have been whether they work or not, has been a bit controversial, although a big European trial has been starting in the last year or two. And likewise with the heart, the animal experiments look fine, but you need a hell of a lot of cells to repair a heart, and it's not so simple as just injecting them. So I thought I would show you, as an example of this, the treatment for age-related macular degeneration. So go back to school biology and look at the layers of the eye, and you'll all remember that on the inside of your eye is the retina, which is full of these photoreceptors. And you might not remember that underneath the photoreceptors is a black layer called the retinal pigment epithelium. And that turns out to be rather more important than you might think, particularly in the context of this age-related macular degeneration. Now, in the middle of your eye, there's a very dense area of photoreceptors called the macula, and this is responsible for the most acute vision in the center of your visual field. But for a lot of us, as we get on a bit, the macula starts to degenerate, and this has really quite serious effects on people's vision. And you can see a case here. This is looking through the pupil at somebody's retina, and you can see a lot of debris and junk in the central region here. Well, this is dry macular degeneration, which is the majority form of this condition. And it doesn't lead to absolutely total blindness, but it can certainly make your vision very bad and make it impossible to do anything. And it's relatively common. You can see these figures in the UK. Quite significant numbers of people are suffering from this. And this is actually due, or to a large extent due to faults, failures, disease in the retinal pigment epithelium layer. And if you look at the normal pigment epithelium, it looks like this. And each of these little green things is one cell. And if you look at a macular degeneration retinal epithelium, it looks like this. You can see the cells are very irregular, and some of them are fused together. There are gaps and so on. And it turns out the retinal pigment epithelium does quite a lot of things that are very important in the eye. So here's another picture. Here's the photoreceptors. Here's the pigment epithelium, which for some reason is colored white here and not black. That sits on a basement membrane, and underneath is a layer called the choroid that's got a lot of blood vessels in it. And number one, the pigment epithelium helps recycle the visual pigment that reacts to photons when they hit your receptors. And the visual pigment has to be cycled out of the photoreceptors into the pigment epithelium and back again as it's resynthesized. Secondly, it's important, phagocytosis means taking up debris, absorbing bits and pieces, and it keeps the retina clean by, again, as the photoreceptors are working, they're constantly shedding bits and pieces, and the pigment epithelium will clean those up and get rid of them and avoid obscurity or degeneration resulting from too much of this debris. And it's also supposed to control the angiogenesis, although I guess that's more relevant to the wet type of macular degeneration, which is due to proliferation of blood vessels. And well, there's a long history of biomedical research on this condition. But one of the key parts of it was something called the Royal College of Surgeons rat, which is a line of rats which has spontaneous macular degeneration. And it was found, 20, 30 years ago, that if you can graft human retinal pigment epithelium under the photoreceptors of the RCS rat, and it would repair the damage and improve the vision. So for some time, people have been thinking this would be a great application for pluripotent stem cells. And also it turns out that, in fact, in this particular case, the differentiation protocol for making pigment epithelium is amazingly simple. And in this method is still used today. Here's your embryonic stem cells or iPS cells. If you take away some of the factors in their medium, they'll clump into things called embryoid bodies, which are sort of mangled mishmashes of early cell types that the embryo normally forms, but without very much spatial structure. And then as though by magic, if you just leave these sitting around for a week or two, a number of them will go black. And those embryoid bodies are producing retinal pigment cells. And you pick out the black ones and you replate them, and then you get more black and more black until it's 100% black. And then you've got a nice culture of retinal pigment epithelial cells. This is transmitted light, and you can see the cells are all got some black grains in them, which is the melanin pigment. And this is the same sort of view that you saw previously of the cell junctions. And actually, these are gaps. These are not malformed cells. These cells look very nice. And there have been quite a lot of clinical trials involving grafting this RPE underneath the macular region of patients with age-specific macular degeneration. And the consensus now seems to be it's better to actually put the cells on a substrate and graft this in rather than just injecting the cells. So this is a recent example of this a couple of years ago. Here are the cells, and these are put onto a sort of plastic or parylene. This is a massively magnified picture to show you that the membrane has holes in it to let both cells and substances through. This is the implant coated with cells. It's about a few millimeters long. Here is a patient with debris in the macular region. This is after grafting. That's the size of this implant in the eye. And this is the same thing, but looking using fancy computed optical tomography that ophthalmologists use. And this particular trial has these results. So fairly small number of patients using embryonic stem cell derived RPE, grafted on a membrane, about 100,000 cells, which is not very many cells, just a small dish of cells. Interestingly, because this is the eye, the immune reactions against graft are much less than they are in most other parts of the body. And I think the reasons for that are not understood perhaps quite as well as we would like, but it is the case that surgery on the eye or transplants into the eye seem to require modest amounts of immunosuppression. So this is quite modest. There's a bit of steroid and some tacrolimus, which is a standard immunorejection drug, tapered off after a couple of months. Side effects are not too bad. Now, this particular group, they were all very badly affected by their disease, and so it was difficult to see whether they were getting better, but a few of them got a bit better. And that does seem to be the typical result for these phase 1 trials, that you don't get sort of 12 out of 12 leaping out of their chairs and reading the last row on the optician's chart, but some of them seem to get a bit better. And this probably does work. And when they start treating less seriously ill people on a larger scale, I think that will become more clear-cut. And just before I finish with pluripotent stem cells, I just point out that this particular application does require a very small number of cells. And that's also true for Parkinson's disease. But if you go to the other extreme of heart failure, you're needing a 10 to the 10 cells, which is really a lot, a big flask of cells, and quite an effort to grow that many clinical grade cells under extremely tightly controlled conditions. Okay, let's move on to tissue-specific stem cells. And these are the normal precursors to each tissue type in your bodies. But many people think there are stem cells everywhere, all over your body, but there aren't. Some cell types don't divide at all from the day you're born to the day you die. For example, the corneal endothelium, the lower layer of the cornea, cells don't divide at all. You can make them divide if you put them in culture, but in vivo they never divide. And there are many tissues in the body that divide a little bit, but don't really have an identifiable stem cell population. But some tissues do have stem cells, and these are particularly well characterized examples. The lining of the intestine constantly turning over, fed from a population of stem cells in the crypts. The epidermis of your skin, you all know that your skin is constantly falling off, and you're constantly growing it from the basal layer. What I should talk about for a few minutes now, the blood and the immune system, very important for all sorts of reasons. And for those of us with Y chromosomes in the audience, we also have some very important stem cells down below producing sperm. But all of these tissues, just a few randomly selected examples to which is pretty clear there are no stem cells. Sometimes there's a bit of cell division during life, and the heart muscle, for example. People have wanted there to be stem cells in the heart so badly, and they've really tried very hard to find them. But the best anybody's been able to do is to find that the heart muscle cells divide a little bit, but on average, it's about half a cell division per cell in the whole heart in a whole lifetime. So that's not stem cells. Right, so let's have a look at the blood and immune system. This is fed by cells called hematopoietic stem cell. Hemo for blood and poiesis, I believe, is Greek for something or other. My wife will tell you more about that. And every textbook of hematology or developmental biology will contain a diagram like this. And this is the third edition of my textbook. Actually, in the fourth edition, I've taken it out because it's not wrong, but it's over prescriptive. There's too much detail on this, which is not really sustained by recent data. But I'm leaving it up here for tonight because it's easy to explain the importance of the stem cells and how they relate to red blood cell production. So here's the stem cell population renewing itself, and this feeds a whole load of different cell lineages, one of which leads to the red cells, which, by the way, involves the nucleus being ejected at a late stage of maturation. Another lineage gives you blood platelets. Another one gives you some things called neutrophils, which are important for non-specific immunity and all sorts of other things. Macrophages, which go around scooping up debris, and they're all important lymphocytes that carry out all the functions of your immune system. And the respect in which this diagram is not quite as precise as one might like is that these cell types up here, all the ones that aren't the ultimate stem cell, are probably all the same. They're one sort of mass of rather similar non-committed cells. But you don't need to worry about that tonight. And the hematopoietic stem cells are real. I mean, they were postulated many decades ago, and then bone marrow transplantations made people think that they must exist, and then they were isolated first in mice and later in humans, what, about 25 years ago now in humans. But now you can actually see them, and this is a mouse. This is a very fancy, rather clever experiment in which an extremely complicated method of genetic labeling has been used to make the stem cells green. And they're actually imaged in a live mouse in the calvaria. That's bones of the marrow of the calvaria, which are bones in the skull. And the mouse has been shaved, and a very fancy microscope called a 2-photon microscope has been pressed against its skull. And that can see deep enough into the tissue to be able to image these cells. So these really are hematopoietic stem cells. And the main point of this paper was to show that they're all stuck to this blood vessel, which is shown here in red. And it does seem to be that the stem cells like to adhere to blood vessels in the bone marrow, and that's their so-called niche, which they need to keep on behaving as stem cells. So hematopoietic stem cells are super important from a transplantation point of view. And in fact, transplants of hematopoietic cells are virtually all the stem cell therapy there is. Well, they are all the stem cell therapy there was from the 1980s to about 10 years ago, and they're nearly all that there is today. Really very important. So, and I'll explain why in a moment. So the cells come, as the name suggests, from the bone marrow, but increasingly, and particularly in Europe, as opposed to the States, from peripheral blood, because there are various substances now known that can mobilize them out of the bone marrow, and they can be enriched or partially purified from blood. And also umbilical cord blood can be a useful source. So those placentas that used to be thrown away contain fetal blood with a very high proportion of hematopoietic stem cells in it. And bear in mind for the next few slides that there are two very, very different sorts of hematopoietic stem cell graft. There's the autologous graft, which is a graft of your own bone marrow, and I'll explain why that's important in a moment. And there's the allogeneic graft, where it's somebody else's bone marrow. So these are the main bullet points. And we call it hematopoietic rather than bone marrow now, because the cells don't always come from the bone marrow. Overwhelmingly the most important type of stem cell therapy. The numbers have been going up steadily, and it's about 100,000 a year worldwide now, mostly in North America, Europe, and Far East. And they're mostly for treatment of leukemia and lymphoma, and some for genetic diseases of the blood, which I'll come on to in a minute. Now, why are they used for treatment of these cancers of the blood? Well, the idea came about in the 1950s because, it was nuclear weapons, nuclear power development time. There were radiation studies on animals. There were radiation accidents affecting people. And it was known that animals that had received lethal doses of radiation could be brought back to life by giving them a bone marrow transplant, just intravenously. And that got people thinking. They thought, oh, there must be cells in the bone marrow which are killed by radiation, but which will repopulate an irradiated host. And maybe they thought they knew they could kill leukemias and lymphomas with radiation or with, actually at the time, rather primitive chemotherapy. But they also knew that if they gave enough to kill the tumor, they'd also kill the patient stone dead because their own bone marrow would be destroyed. But then they thought, well, supposing we give them a lethal dose and then we give them a graft of somebody else's bone marrow, then they'll be brought back to life and they'll be good as new. And we know from these animal experiments that the graft can repopulate the host and generate new blood for life. Unfortunately, the first experiments were not very successful. And in the 50s, all the patients died from treatment-associated complications soon after the grafts. They went a bit out of fashion in the 60s, and back in the 70s, there were one or two successful results. But it wasn't really till the 80s that it started to work properly. And I think it is universally believed by stem cell biologists now that if the sort of regulatory controls were enforced back in the 50s that are enforced now, that bone marrow transplants would never have been invented because they're so dangerous. Well, I've heard it said that the first 60 patients that were treated all died, virtually all of effects of the treatment. Now, you could not possibly do that nowadays, and it is a food for thought. And I haven't mentioned the last bullet point yet, because what I've said so far was the original rationale. But by the 70s, it was becoming clear that the graft itself was doing something. It wasn't just repopulating the blood, it was actually helping to kill the tumor as well. Of course, it has to be an allogeneic graft to do that. It has to be somebody else's bone marrow. So here are the numbers. Autologous grafts use, this is figures for Europe, so you can magnify this up and get your 100,000 for the whole world. Very few 1990, it was just beginning to work then, but steady increase. The majority are actually autologous grafts and the minority are allogeneic. And the autologous graft, I made fun of injecting bone marrow right at the beginning. You may think, what's the point of an autologous graft? The point is that it is your own cells. And so if you do this procedure of irradiating or giving chemotherapy to the patient to a lethal degree, you could theoretically save them by taking out their own bone marrow, putting it in culture for a few hours, giving them the treatment, and then putting the bone marrow back. The reason why that isn't done all the time is because leukemia is a disseminated tumor and the cancer cells are all over the body, including riddling the bone marrow. So there would be no point trying to do this for a leukemia. For a leukemia, it has to be an allogeneic graft from another donor. If it's a lymphoma, though, lymphomas tend to be more lumps of tumor. Not always, they can be disseminated, but on the whole, it's thought more reasonable to use autologous grafts for lymphomas because there's a better chance of getting a sample of bone marrow from the patient which doesn't have cancer in it. So anyway, they are both used and they're both increasing in popularity. So I've already hinted that allogeneic grafts are not very good for you. And the reason is it's this phenomenon called graft versus host disease. Now, you've all heard of graft rejection. If you have a heart transplant or kidney transplant or something, the host tends to reject the transplant, and it does it because something called the HLA system recognizes non-self in the graft, and your own lymphocytes attack it in all sorts of complicated ways. Now, the graft rejection is not so much of a problem for hematopoietic grafts because the graft can only survive in the patient if the patient's had his own marrow destroyed. And if your marrow has been destroyed, you don't have much of an immune system to reject the graft. So graft rejection is not so much of a problem. But of course, the graft itself is either full of lymphocytes, or even if it's purified stem cells, it's going to produce lots of lymphocytes fairly quickly. And if it's an allogeneic graft, they're going to be lymphocytes which recognize the host as being foreign tissue and attack it. And that can be very serious. In fact, it can be easily lethal. So allogeneic grafts, on the whole, one of the things, there were three things really that made this procedure work rather than killing everybody. And one of them was the discovery of tissue typing, which is based on this genetic system called HLA, human leukocyte antigen. This is all very complicated, but you all know that everybody's different, that there are widespread differences between people, but you can match people, and if you've got there's usually about 4 to 8 different genetic loci will be matched, and you want as many matched as possible for an allogeneic bone marrow graft. Secondly, there was the discovery of immunosuppressive drugs, which are very sophisticated now, and there are several different classes of these, but the most widely used, I guess, are the ones that stop T cells, that's T lymphocytes, producing a substance called interleukin-2 that they need to reproduce themselves and build up numbers to become very dangerous. So immunosuppression reduces the division of these lymphocytes. And also there's a lot of very fancy antibiotic treatments for the infections that people invariably get if they are suffering from GVHD, and also very sophisticated intensive nursing methods have also improved a great deal. So for these reasons, quite a lot of people who get allogeneic grafts do survive and are indeed cured now, but there's still a significant mortality, and it's still very dangerous. And actually, I can never get a hematologist to give me a real figure for mortality because it all depends on exact type of disease and the type of graft and this and that and the other. But, figures of 10%, 20% seem to be still the case, at least in some circumstances. So that's a very quick indication of the use of hematopoietic cell grafts to treat lymphomas and leukemias. And in my undergrad, I do still give a few undergraduate lectures on this subject, and I like to describe to them some of the research activity that's some of the frontier research in this area. And there are quite a lot of different things actually going on. But I thought the one I'd show you would be gene therapy, because the property of the hematopoietic graft, that you can replace somebody's blood and immune system for life with donor cells, means that this is a terrific opportunity to get genes into the body. You've all read about gene therapy, and you all know it's been very slow to be developed. And the main reason is it's very difficult to get genes into the target tissue. So if you want to get genes into the lung epithelium to treat cystic fibrosis, you've got to get these viral vectors down the bronchi into the cells. Really difficult, very low efficiency. Problems haven't really been solved even now. But if you can get your cells in culture, then you can do things, whatever you like to them, and then you can put them back into the body using a standard clinical grafting procedure. Then that's another matter. And it's always been apparent that hematopoietic grafts would be useful for treating genetic diseases of the blood, because obviously if you give a graft from a healthy person and replace the bone marrow of somebody with disease, then you've fixed the problem. And in recent years, last 10 years or so, there's been a lot of attention given to sickle cell disease and thalassemias from the gene therapy point of view. So you've heard of both of these diseases. Sickle cell disease is a mutation of hemoglobin beta. So hemoglobin is the red pigment in your blood that carries oxygen. There are several hemoglobin proteins. In adults, one molecule is made of 2 alpha subunits and 2 beta subunits. And there's this rather common mutation of hemoglobin beta known for some time, which prevents it forming the normal molecular structure of hemoglobin and makes it polymerize. And for some reason, the cells containing a lot of these hemoglobin beta polymers can develop this sickle shape. And that means they block up small blood vessels when they try to go through them. And you get terrible organ damage and terrible pain resulting from this sort of problem. So sickle cell disease is really a very unpleasant condition, mostly because of this blockage of blood vessels. And the thalassemias are a collection, a group of different genetic diseases, also of hemoglobin beta, but in different parts of the protein molecule. And they all have the effect of reducing the function of the hemoglobin beta, either preventing it being made altogether or making a protein of reduced function or less efficient function than usual. And this means your blood's just not working very well, and you tend to need a lot of blood transfusions, and then you get all sorts of problems like iron overload arising from the transfusions. And the cells don't form sickles, but they can go a bit out of shape, as shown here. Now, both of these diseases, sickle cell and the thalassemias, are most common in sub-Saharan Africa and the Middle East, and to some extent the Far East. They're all areas where malaria has been very prevalent for a long time. And the reason these diseases are so common, despite being so serious, is because the heterozygotes, the people with one good copy and one bad copy, are more resistant to malaria than normal people. So one copy of the mutation helps you survive malaria, but 2 copies gives you one of these diseases. So you're all thinking, well, why can't you just fix these with an allogeneic graft? You know, why bother with gene therapy? Why not just take somebody's stem cells and graft them in? And that has been done, and it does work. The problem is that for the reasons I've just explained, it's very, very dangerous. And unlike somebody with leukemia, who may very well be dead in a year or a few months, these people, although the disease is bad, it's not quite that bad, and they probably would survive for decades with disease, but they're not going to die in the next year, probably. And it turns out, this is actually a big meta-analysis that was done a few years ago of allogeneic grafts for, this is for sickle cell disease. And it turns out that the blue line is basically what you want, but this is a completely HLA matched sibling. So, 8 out of 8 or 10 out of 10, or however many alleles you're doing from a sibling. And the siblings are better than strangers with an HLA match because they've also got lots of other genetic loci, which, 50% of all their other loci will be matched as well. So if you've got an HLA matched sibling and you're under 13, and so able to take the tremendous rigors of this sort of treatment, then the overall survival is very good, and the event-free survival, that's no ghastly episodes of pain, is also, that's sort of 95%, and this is almost 100%. So that's very good. But you've only got to go down to the that haploidentical relative. That's basically a sort of 50% match. A stranger with a good HLA match, or even worse, an incomplete match from a stranger. And you're getting down to areas, you're getting sort of 10% mortality here, and that's just not on for this type of patient. So the indication for the allogeneic graft is only for a very few people who are fortunate enough to have a complete matched sibling. And the smaller families get, the less likely that's going to be. So that's why where the gene therapy comes in. And the good thing about both of these diseases is that if you provide enough of the good copy of hemoglobin beta, then it can compete out the mutant form, and you form productive dimers. And actually, there is a single base substitution artificial form of hemoglobin beta that's even better at competing out the mutant form, and that's what's normally used for gene therapy. So here's the hematopoietic diagram again. Here are the stem cells. This is the red cell lineage. And this is the sort of viral vector that's used to put genes into the isolated stem cells. So just very briefly, these bits at the end are to do with the virus, and forget about them. These three gray bits are the actual bits of coding region for the good copy of the hemoglobin beta. And then that's a beta hemoglobin specific promoter. And this is what's called an enhancer. It's a regulatory region of DNA. Now, these are important because they make sure that the gene is only expressed in this lineage, only in the red blood cell forming lineage. You don't want lots of hemoglobin being made in all these other cell types. And that will cause all sorts of other trouble. So there's obviously a lot of careful design involved in these gene therapy vectors. And I wondered whether to tell you about pseudotype lentiviruses, but I've decided not to. And here's a trial done. There was one actually in the news just recently on the BBC, but I couldn't find the actual clinical trial data for that. And also I noted that it was a slightly different sort of gene therapy. It's a bit more complicated than this. But this illustrates exactly what I showed you on the previous slide. Small trial of 7 patients, all with beta thalassemia. And they were given, I won't go back, but anyway, the gene therapy vector you've just seen, the good copy of hemoglobin beta. Here are their erythroblasts. That's the cells that are going to become red blood cells. In the diseased marrow, they look a bit grotty. And here's in the patient 4, which is the one that gives the best results. They look pretty normal. And these are the 7 patients. Now, these are expressed in terms of how many blood transfusions they're getting, because that's the treatment for thalassemia. And before the gene therapy, they're all getting lots and lots of blood transfusions. And after the gene therapy, there are 2 years time courses here. 3 of them, which were all children, as it turns out, are completely transfusion free. So they are 100% cured, probably for life. 2 of the adults are much lower, about half as many transfusions as they used to have. And 2 of them are not very good. They're really back to where they were. Now, why the variation? It's because of the proportion of the hematopoietic stem cells in the marrow that you managed to reprogram. And if you get most of, as you probably only need to get about half of them, and then you'll get a cure. But if you're only getting 10% or so, then you may get a bit of an effect, but it won't be good enough to be a permanent cure. So it's all a numbers game. You've got to get as many stem cells as you can. You've got to get as many genes into as many of those as you can, and you've got to get good repopulation when you put the cells back. And this is the last slide, fortunately. Some people might be thinking, this is all very expensive. How can the NHS possibly afford it? And well, you can discuss this for years, but I just thought the gene therapy that was mentioned for thalassemia on the news the other day cost about £2 million per treatment. Hello, there's feedback suddenly started. Does anybody know why that's happening? Maybe that's a signal that I should stop. Can you do anything about the feedback? Okay. Well, a regular allogeneic graft, as you can see, I shouldn't say much, but you can all read this. All is of adjusted years, and this is actually value, pure and lymphocytic leukemia.
The should be muted.
You happy to answer some questions?
Well, if we've got rid of, yeah, the feedback has gone, hasn't it? Okay, right. Mhm.
Yes, I'm interested in non-relevance of stem cells to MS.
It started again. Is it that microphone that's the problem?
Maybe we just
Okay, the question, for the benefit of people on Zoom, the question was relevance of stem cells to MS. Well, somebody who advertised this talk on the email mentioned this recent news story about MS. So I did look it up actually, and it's I think, to be fair, it's a reputable scientific study. It's using it's sort of in the tissue-specific stem cell category. It's using neural stem cells, a cell line of neural stem cells, and grafting them into the cerebral, into the cerebrospinal fluid of MS sufferers. To be honest, all that the trial says is that the treatment is tolerated, and that there are no serious adverse effects, and no problems with the cells becoming tumorous or anything. So it's really a phase 1 trial. Some of the news reports have rather exaggerated the significance and suggested some of the patients might be getting better, or even that the degree of inflammation in the brain might be reduced. Now, actually, the paper does not claim any reduction of inflammation, although some of their mouse experiments do show that. So I would say this was an extremely early piece of work.
try.
Hello.
It's okay, the lights on.
Hi. I thank you very much. It was very interesting, particularly for me, because my cousin had leukemia and had the bone marrow transplant from his brother. Unfortunately not successful, but I now understand what that was all about. But my particular interest is in Parkinson's, which you did mention, and I didn't quite understand what the stem cell was doing, because Parkinson's is mostly about dopamine. Is it replacing the dopamine cells?
Yes, well, there's this population of cells in a bit of your base of your brain called the substantia nigra. Contains a concentration of cells which use dopamine as their neurotransmitter. And these cells die off, or may quite often die off, in old age. And that's what causes Parkinson's disease. And so you get these movement problems, and it tends to be progressive. There are drugs to treat it, which really are stimulating production of dopamine, or facilitating, encouraging the activity of the dopamine, and these have some value. The idea of the stem cell therapy is that you get some pluripotent stem cells, you make the dopamine cells, dopamine secreting neurons in vitro by, the sufficient knowledge of embryology and putting the right substances on, and you graft those into it's usually into the target tissue, target region of the neurons in the brain, so not actually into the substantia nigra. And the idea is you're replacing the cells that have died off, so that really would be proper stem cell therapy. Now, this actually started not with pluripotent stem cells, but with dopaminergic cells taken from aborted fetuses, from the fetal midbrain. And there was some work in Sweden and later some work in the US, grafting these cells from the fetal midbrains into Parkinson's patients. And the Swedish data was fairly positive, and they had movies of people, walking steadily up and down the hospital corridor, whereas previously they'd been falling around. The American study wasn't so convinced and didn't think there was much effect at all. Then there was a lot of argument about had they used quite the right cells, and had they put it quite in the right place, and had they used the right number of cells and so on. And I think actually the consensus seems to be that it probably can work, but nobody's quite pinned down exactly what you've got to do. And at the same time, of course, particularly in the US, the idea that you're going to routinely use tissue from aborted fetuses for therapy doesn't go down very well. So this has had a new lease of life from the pluripotent stem cells because, well, even if you don't like human embryonic stem cells, you'll be willing to make them out of iPS cells. Last time I looked this up, there was a big multi-center trial starting in Europe using actually fetal midbrain and ES cell derived and iPS cell derived dopaminergic grafts. So I think it probably will work, I must say. That's my gut instinct, but I don't think it's been proved yet.
Thank you. That was very. I thought that was very interesting. I also looked up the study of the stem cells in MS. The claims of improvement were actually a sort of lack of deterioration. And one of the striking things for me is it wasn't a controlled study, and all the patients received quite a lot of significant immunosuppression. Yeah. And it's scientifically plausible any benefit would be from the immunosuppression. Yeah. And that makes it quite hard to do a controlled study unless you're giving immunosuppression to patients without the stem cells. Yeah, I know you're not a clinician. I actually spent some time in the mid-80s and early 90s doing bone marrow transplants on children for metabolic diseases. So just to add to what you were saying is some of the early transplants were done for patients with severe immunodeficiencies. So the risks to them were much lower because you didn't need to give them immunosuppression. And we did used to counsel the families about a 10% mortality. That was the figure that we would give them. Yeah.
Yeah. Well, yes. Actually, I don't know. Is there any way we can find out whether the Zoom audience, can they hear the roving microphone?
They do.
They do. Okay. Well, I hope they heard that interesting contribution. Yes, I mean, metabolic diseases, that's another sort of area of gene therapy where this type of graft can be very useful. Because supposing you're lacking an enzyme for genetic disease, it doesn't really matter necessarily where the enzyme is normally located in the body. If what it's doing is removing something from the blood, and in the absence of the enzyme, some toxic product builds up, because that's quite a common scenario, then you could put the gene for that enzyme into hematopoietic stem cells, put them into the patient, and now it's expressed in the blood. And even if it was normally expressed in the liver, it doesn't matter because the blood can go everywhere and it can have the same beneficial effect and get rid of the toxic accumulation. So there's a whole load of metabolic diseases where this is potentially a very useful way of method of gene therapy. So yeah, that's just a general addendum to what you said.
Okay, I've got a question. I was part of a stem cell trial in Bristol for MS. So I received, my own stem cells from my bone marrow. Now, I certainly didn't feel any different. And the study at best was probably inconclusive. Do you have any suggestions for alternatives that I should be exploring?
I don't think it's my place to advise people on treatments. I'm not a clinician. Okay. I would just say one thing though about why do people believe in stem cell therapy when there's, in those cases where the evidence is rather dodgy. Well, obviously everybody likes to believe in new cures, but quite often it's pretty clear that, say, the director of some private clinic in some unnamed country selling stem cell injections, he has a range of people coming to him with different conditions. And a lot of them may come from abroad and go away again. And he'll inject the cells, and the people have a range of diseases. Sometimes they'll feel better, sometimes they'll feel worse afterwards, sometimes they'll get better, sometimes they'll get worse. But on the whole, the ones who feel better write letters, or these days emails, saying, your stem cells are really great and I've been cured. And then these letters are testimonials that go on the website of this clinic. Whereas the ones who've died or got worse probably don't write and say how great they feel, and they are ignored. So this is a sort of reporting bias, which is very familiar to medical statisticians for, donkey's years. And it arises when you don't properly control the trials. And of course, trials are expensive and difficult to set up, and, lots of money, lots of organization, and can be very, very difficult to control. And, the previous speaker mentioned that you can't do a trial where all your placebo group are immunosuppressed, because the immunosuppressive treatment is pretty aggressive, and it wouldn't be ethical to give people that when you know that they're not getting anything, that it's for no reason. So yes, there are all sorts of beliefs out there which grow from understandable sociological reasons, but not necessarily good scientific reasons.
Thank you. A comment, well, statement actually. Success in science is often related to luck. What do you see in terms of the next big breakthrough in stem cell biology? And where do you think we're going to be in 10 years' time? I like that question.
Well, whenever any basic scientist is interviewed by a TV or newspaper and they're always asked, when is your discovery going to be in the clinic? And they always say 10 years, because that seems like a long period of time. And by 10 years, everybody will have forgotten about my promises. I think actually there's an awful lot of interesting stuff going on with hematopoietic stem cells. So, for example, there's some mouse experiments I saw a year or two ago which show that if you purify your stem cells to a high degree and have lots of them, many more than in a normal graft, then you can repopulate a host mouse without having to irradiate it or without having to give it a lethal dose of radiation. Now, that's an enormous game changer. If you could amplify those HSCs in vitro for human HSCs and make the transplants so much safer, because you don't have to give the lethal doses of chemotherapy or radiation, that would enable loads more things to be treated. Lots of autoimmune diseases, for example, which would respond to replacing your immune system. So I think that's very important. There's another HSC-related phenomenon which has been around for a while, but seems like it ought to be important. It's been known for a long time that if you given a successful allogeneic graft, then the host is tolerant to subsequent solid organ grafts from the same donor. And so the idea has been around for a long time. If you want to have an immunosuppression-free heart transplant, why not do the bone marrow transplant first, let it settle down, and then do the heart transplant? And well, the reason is because the bone marrow transplants are too dangerous. But supposing they were safer, then that would effectively render people tolerant to foreign solid organ grafts, at least from one particular donor. And if you think it's difficult having blood and a heart from the same donor, well, you can make iPS cells from the organ donor at the time you harvest the heart. So there's all sorts of things like this that could happen, which would be really game-changing. That's enough speculation. Anyway, you can imagine all these things as well as I can.
We have got another question online. Did the grafting of the bone marrow result in death because of rejection?
Well, it's rejection in reverse. It's usually the graft versus host disease. So it's the rejection of the host by the lymphocytes in the graft. And that causes all sorts of damage to and all sorts of infections.
I've got another one online, but until you find, does anybody in the room? Maybe we'll just alternate.
I was just going to comment, with old-fashioned bone marrow transplants, the risk of failure of engraftment was relatively low, about 2 to 5%, but it was very serious and potentially fatal if it did occur.
Yes, indeed.
I've got a question here from Duncan. If the initial course of treatment fails, is it feasible at any point in repeating the treatment, and is there any point doing it?
Well, I know, again, I'm not a clinician. I do know that allogeneic grafts are not a first-line treatment for leukemia. They're usually used if the chemotherapies failed. I don't know if repeated bone marrow transplant would be attempted. Maybe our colleague here can advise on that.
I didn't really work very much in terms of leukemia and lymphoma transplants, but there are patients who have had multiple autologous and even multiple allografts in an attempt to do that, but it's not very common.
My husband had leukemia and he had a bone marrow transplant, and 15 years later it presented as myeloid dysplasia. So then he went to King's last September for 2 months and had T-cell infusion, which unfortunately didn't work, and he died in January. But the T cells were millions of them put into his body. He then got VRE as well from the hospital, I think, and a fungal lung infection and goodness knows what. So with hindsight, we wouldn't have done it, but he gave it his best shot. So
yeah, well, I can't really comment on that, except it is life and death business.
yes.
I think we'll draw this to a close now, and I'd like you to join me in thanking Jonathan Slack very much for a very interesting talk.